Limited, China, and the presentation title is Application of Bile Acid in Aquatic Animals.
So a very good day to everybody. To Sarah. Sorry, just now my internet got some problem. Sorry about that. And thanks for your
no problem.
Thanks for your give us the chance to share some knowledge on bile acid to global friend. Okay, so I will start my presentation now. Hey, hello, Sarah.
Okay. Yeah, hello.
Yeah.
Let's share the screen here. Okay.
The first. So my topic will be application of bile acid in aquatic animals, and hopefully this will be help our farmers from all over the world. Okay, this is a talking point. First, I will generally introduce some knowledge on bile acid. I believe some of friends, when they first heard the bile acid, they thought maybe this is acid, but definitely it's not acid. Later I will explain. And the second part will be function of bile acid, and the last one will be application in aquatic animals. Okay, bile acid actually is a main active component of bile. It's synthesized in the liver cells using cholesterol as a precursor, and also now we can artificially synthesize in our factory. Okay, the main function is the first is protecting the liver and the gallbladder of fish and hepatopancreas of shrimp, and also can promoting lipid digestion, absorption, and metabolism. Okay, so actually now in the market there are a lot of commercial products for the human being. This is a commercial product in China company for human, which is a cholic acid capsule, which is a CDC. Of course, in the aquaculture, we also use this kind of product. Okay, for the bile acid, actually have free bile acid and conjugated. For the free bile acid, such as cholic acid, deoxycholic acid, and cholic acid, cholic acid, this is free bile acid is less than 10%. And the mean is conjugated, more than 90%, which is free bile acid combined with glycine and taurine, like GCA and taurocolic acid TCA, and glycodeoxycholic acid GDC, and so on. This 6 is a mean conjugated bile acid. And for the function of bile acid, especially for the lipid emulsification, digestion, and absorption. For the bile acid, actually is one of products which can emulsify the fat globule into the micron droplet, fat droplet. Okay, first cause the big fat globules to break into the microscopic droplet. And but remember, emulsion is not digestion. This is only increase the surface area of fat drop and make the lipids easier to digest the fat. And as we all know, lipids cannot access inside of lipid drop, only surrounding the droplet. Okay, this is a cholic acid. Actually, later I will explain. Actually, cholic acid have two parts. One is the hydrophobic side, and another is hydrophilic. Okay, the big fat globule and combined with bile salt, and finally emulsify to droplet. And the second function for the bile acid is digestion. Actually, after emulsion, we can see this photo, triglyceride surrounding by bile salt. And the function for bile acid, they can activate lipids. Okay, this is a lipid, this is bile salt, and break the lipid space structures. Finally, form the hydrophobic channel that to digest the fat. I mean the microscopic droplet. Okay, actually from 1980, there are plenty of research on bile acid which increase the lipid activity. I think from Japan, from China, and even some European country. The animals on eel, frog, turbot, perch, grass carp, and tilapia. If you are interested, I think we can share the whole papers to our audience. Okay, the last step is for the absorption. Actually, after emulsion, digestion from the mast cells, which is fatty acid and bile salt, we can see here. Finally, from the mast cells. Okay, actually the fat cannot dissolve in the water, but bile salt have two parts. Just now I mentioned already, which is a hydrophobic and hydrophilic side. So this one can attract to both fat and water, so carrying the mast cells close to absolute cell, then finally detach. Then for the short and medium chain fatty acid absorbed by capillary and back to the blood. And for the long chain fatty acid, they form into triglyceride. Triglyceride then transported in kilomicrons into lymph vessels and back to the recirculation. Okay, this is for the digestion. And also now we found that bile acid interact with gut microflora. Okay, this is the microflora in the gut. Okay, and can reprocessing the bile acid pool and the regulation of bile acid metabolism by bacteria. So this is for the human being do a lot of research, but for the aquatic animals, short of research. I think in future, maybe more and more research institute in university, even company, they will do some such kind of jobs. And let me do the quick summary. Of course, bile acid can promote the absorption of fat and fat-soluble vitamins, stimulate the intestine to secrete digestive juice and peristalsis, and so on. And second is free bile acid. Free bile acid can inhibit the growth of intestine bacteria, maintain the barrier function of mucosa and the stability of the intestinal environment. As we all know, the drug cannot go in access blood-brain barrier, but bile acid can. Now, for the human being, they have such kind of evidence. And the third part is for application in aquatic animals, but actually for the poultry, for the ruminant, for the swan, they also using quite common in China. Okay, actually, I just share a few papers and research have been done in the aquatic. If you Google, you can get plenty of research on this. Okay, the first is on sea bream fed with the soybean meal. As we all know, in the aquaculture industry, many researchers and more companies are trying to replace the fish meal with the plant protein, especially the soybean product, like soya protein concentrate. Okay, for this trial, for the control is fish meal, totally fish meal. For the treatment, we replace 53% of soybean concentrate and add 5% fish meal. But finally, we found, okay, fed with the high percentage of soybean meal, the body weight of the fish dropped, decreased by almost 20%. Feed efficiency decreased by the 16%, and the daily feed intake increased. Okay, of course, the mortality also increased. And we also get the result is on concentration of bile salt. Okay, dropped, decreased by 10%. And for the cholesterol, as we all know, for the plant protein is short of cholesterol. So that's why the cholesterol dropped almost around 24%. That means when we replace the fish meal with the plant protein, maybe the linseed, maybe the other soybean, maybe we need to add some bioacid. Of course, we are doing the trials, the research on this area. We are trying to find out, but the trial is on halfway. Definitely, we found if replace the fish meal, I mean if the feed add more than 40% soybean protein, soya protein, if don't add bioacid, the growth rate will drop, which is similar to this one. Okay, we will optimize the concentration, and so many jobs need to be done. And of course, just now we mentioned bioacid can protect the liver, gallbladder, improve the FCR, which is a similar feed efficiency, and also promote growth rate. This is done by the other researchers from China, one of universities. When we add 300 gram bioacid in per ton's feed, so the initial weight is similar, 111, but finally we found after the trial, the final weight increased almost 10%. SGR also increased around 8%, and the FCR dropped 10%. But because this cobia is very hardy, I think this is the reason why the survival rate no different. These two photos we can see this is unhealthy. This liver we call white liver, which is very unhealthy, and a lot of fat in the abdomen when we open the fish. Okay, this is healthy. This is a red liver. It looks very nice and healthy, and also we found less fat in the abdomen, which means the bioacid did help fat digestion in the feed or in the body. Let's move on. And we also the same trials. Finally, they analyzed the fat, the lipid in the muscle, the lipid in the liver, all dropped. But for the whole fish, no much different. I think because the fish is so big and the lipid is so less, maybe this is an error. And also some researchers found bioacid can reduce the virulence of Vibrio, which is parahemolyticus. This is very common in the seawater culture. Usually it will cause the AHPND, which is acute Hepatopancreas&Nutrition diseases for vitamin, especially for the vitamin. If because through these trials, they found reduced virulence. Okay, I think it will be helpful for the shrimp farmers. You can try. Of course, we will repeat this trial soon. Okay. So the following will be protecting shrimp hepatopancreas, because hepatopancreas for shrimp is very, very important for the nutrient storage, digestion, and many, many functions. And I also like to share some global case, because now we export to 45 countries from Europe, from Latin America, South of Asia, actually the whole Asia. This is from Malaysia. This is our model farm. Okay, they use the bioacid in the run tank, which is 500 square meters. Before, always got the wet fishes, and just now I mentioned EMS, but after using this, add some bioacid, it did increase the production of the farm. Okay, this is from Indonesia. Also, the similar, they use the mud pond land with HDP, the similar result. Okay, the happy boss and the healthy shrimp. And this from Thailand. Of course, we have many, many others because the time is limited. I just shared, I think, this tree of global case hopefully can help our farmers. So this is from Thailand, Satong. The boss have many.
Yeah, hello, Dr. Benjamin. Sorry for the interruption, but we cannot hear your voice.
Oh, sorry. Maybe the internet got some problem. 又听不见了。Okay. So from when you cannot hear me, Sarah? Now it's okay. 掉了吗？
没有，没掉。
Hey, Sarah, now it's okay. Can you hear me?
Yeah, we can hear now.
Now it's okay. Sorry, I don't know miss how many slides. Now is 3.4.3. Okay, from the Thailand customer feedback. Now can you hear me?
Yes, we can hear you.
Sorry about that. Maybe a long distance, the internet not stable. And we also use in tiger prawn. You know, in the Asia, we have a lot of tiger prawn farm. And this is for Vietnamese in China. Last year, we produced 1.4 to 1.5 million tons. And the huge market, of course, for the consumption, we have 2 million tons. That means we're still short of 500,000 tons. And let me do a short conclusion. Bioacid added in the artificial feed can promote the growth rate of fish, shrimp, and even the crab. Okay, of course, definitely it will protect the liver and the gallbladder of fish. Bioacid can improve the utilization of lipid in the fish body or in the feed, reduce the body fat deposition, and we have the trials on this part. And when we replace fish meal with the plant protein, definitely if we add some bioacid, definitely will promote the growth of the fish and improve the feed efficiency. I mean, now we found more than 40%. replacement, we need add some bioacid. Okay, this is our model farm, which is in Thailand. I just take 1.5 minutes to share to our audience. If some friends want to know, understand more information on bioacid, that's the contact of us. This is for tiger prawn and some red meat. Yeah. Actually, at the beginning, they seldom change the water, only top up due to the evaporation, due to the discharge of particles, feces, sediment, and the density can reach 200 to 300. Got some problem again. Sorry, got some problem again. The nice place, nice farm. Yeah. Hi, Sarah, can you hear me?
Yes, we can hear you. We are watching the farm.
Thanks.
Yeah, actually it's nice.
Yes, this is I think 100 acres.
Okay.
Yeah.
It was very long.
I almost finished.
Okay.
And,
You started culturing in that farms?
this is our model farm in Thailand. Yeah,
Okay.
we work here with the local farmers. Yeah. I think this is for my presentation. Thanks for all the friends from overseas, from global. Thanks for your attention. Yeah, this is a cooked shrimp. Looks very nice, is it?
Okay.
Yeah. So, any questions for me? Can you hear me?
Yes, we can hear you. So
Yeah.
any questions, please proceed.
Yeah, just ask.
Yeah, actually this farming is for only shrimps, right?
Yeah, for the shrimp.
Not only.
Okay.
Oh.